All right. Well, we're going to go ahead and begin. Welcome, everybody. My name is Lisa DeVetter, and I'll be the moderator of this webinar titled Wax On, Acid Off, Approaches to Cranberry Fruit Improvement, which is part of our VacCAP webinar series. So we're going to start in just a moment, but before we begin, I want to provide a few housekeeping details. Please keep your cameras off and remain unmuted. That helps with the quality of our recording for future listeners. We are going to hold a Q&A session at the end of this webinar. So please keep your questions in the chat and we will move through those questions at the end. And then we also are going to have an email survey distributed at the end of this webinar. So please keep an eye out for it and help us out by completing that survey in a timely manner. So with that, I want to introduce Dr. James or Jim Polashock, who is a research plant pathologist at the Mariachi Center for Blueberry and Cranberry Research and Extension in New Jersey. So welcome, Dr. Polashock. We look forward to your webinar and feel free to begin.
Okay. Thank you, Lisa. I appreciate the invitation and the introduction. Can you see my slides okay?
I confirm I can see your slides. Okay,
great. So I want to get started. Cranberries are kind of a unique crop. So I'd like to start quickly with an introduction into cranberries. What we call cultivated cranberry or large fruited cranberry is Vaccinium macrocarpon. It's in the Ericaceae family, which are a group of acid-loving woody perennials. The family contains about 126 genera and over 4,000 species. Other fruit crops in the genus Vaccinium include blueberries, several different species of blueberry, and lingonberry, which is on the right-hand side here. There's cranberries, blueberries, and lingonberry. Lingonberry is Vaccinium vitis idea. If you go to IKEA, you might be familiar with lingonberries. They have lingonberry juice and things like that. There are also several ornamentals in the Ericaceae, including rhododendron, mountain laurel, azaleas, and some others. Commercial production in the United States has an estimated economic value of $385 million. The US is the largest producer of cranberries in the world, but there's also production in Canada, Chile, and parts of Eastern Europe. In the United States, by far the largest grower of cranberries is Wisconsin, followed by Massachusetts, New Jersey, and Oregon and Washington up here in the Pacific Northwest. The primary products of cranberries are sweetened dried cranberries, what we call SWDs, and juice cocktail. All of the cultivars in cranberry are actually clonal, which means they're vegetatively propagated. Cranberries runner, shown in this picture here, and the runners in the old days were cut or mowed off, established beds, and pressed into new beds where they root pretty easily to establish new beds. But nowadays on the bottom here, rooted cuttings can be purchased commercially from nurseries. So most of the new beds now are planted from rooted cuttings rather than from cutting and mowing established vines. Cranberries, at least in New Jersey, bloom in about June. The flowers are shown here. They're bee pollinated, so bees need to be brought in for good pollination. Bumblebees work the best, but honeybees work as well. The crop develops throughout the year. These are plots in the middle here, and when the fruit ripen, beginning in September through November, usually September, October, the fruit are harvested. Most of harvesting done now is what we call water harvesting, where the floods are actually, the beds are actually flooded. The fruit are knocked off the vines and collected. Usually we use something like oil booms to corral the fruit, and then use conveyors and water jets to move them off the beds where they go for washing and further processing. The berries actually float because if you cut one open, they have these air spaces or locules where the seeds develop. So they're air filled and actually float pretty well. There are dry harvesting methods that are still used mostly for the fresh market, but the entire process market is pretty much water harvested. So my lab is interested in many different traits of cranberry. For example, epicuticular wax, which is the wax on the surface. There's a picture here where you can see the wax is pretty visible on the surface versus fruit that have a little bit less wax. We're also interested in fruit rot resistance. Those are pictures on the bottom, rotten fruit on the left and sound fruit on the right, which are some of the new varieties that the Rutgers breeding program is developing and we are collaborating with. We're interested in the total anthocyanins, which is the red pigments that give the fruit their color, titratable acidity, organic acids, bricks or soluble solids, fruit shape, yield, and what's becoming more important now is environmental stress resistance. And today I'm just going to focus on the ones that are highlighted here: epicuticular wax, which is our wax on, and organic acid, which is our acid off. So the first question is, why do we want wax on? And the short answer is because cranberries really need sunscreen. Sun scald or fruit damage from irradiation, high temperatures and low humidity is becoming more and more common. And we think it's becoming more common because there are a lot of reasons, but one of the reasons is because we have much higher yielding varieties now. And those higher yielding varieties have a lot of fruit that just sits on top of the canopy, so there's no shading, and the fruit literally gets sunburned. In addition to the losses due to directly to sunburn, we also get secondary fungal infections. So even if the sun scald spots are small, the fruit tend to get infected and rot, and then the fruit is subsequently lost to fruit rot. So that's something that we're working on right now. Right now, sun scald is controlled in cranberries primarily by irrigation. Irrigation can be turned on, or is usually turned on when the temperature exceeds 27 degrees C or about 81 degrees Fahrenheit. This cools the fruit, but it's very resource intensive. We have to run the pumps. It uses a lot of water, fuel, etc. And more importantly, it can increase fungal pathogen infection because fungi typically need a wetness period for infection. So keeping the fruit wet for extended periods of time can actually increase fruit rot and fruit infection. In other fruits, sun scald can be partly controlled by avoiding excessive pruning. So you leave vegetation on to help shade the fruit. Shade netting can be used, or applying sunburn suppressants or coatings to the fruit surface. All of those things are not really practical for use in cranberries. Cranberries aren't typically pruned. To shade all the cranberry beds would be difficult and expensive. And of course, then you're also limiting sunlight that the plants need for photosynthesis. And sunburn suppressants are difficult as well, because we can't just apply suppressants, which are usually things like kale and clay, just to the fruit, because you can see in the way the canopy grows here, where the irrigation sprinkler is on, cranberry is a low growing woody vine. And you can't just spray the fruit, you have to spray the whole plant. And then getting those materials on the leaves reduces their photosynthetic rates and things like that. So it's not really desirable. So we asked the question, can surface wax on fruit act as a natural sunscreen? Surface wax, or the wax on the fruit surface, there's two different types. There's epicuticular wax, which forms a highly crystalline structure on the fruit surface and establishes sort of a continuous hydrophobic self-cleaning surface. In this example on the right from Vaccinium myrtillus, which is a European blueberry or bilberry, a paper published by Clavins and Clavins in 1920 shows in this SEM the crystalline wax structure on the surface of the fruit. There's also what we call intracuticular wax, which is embedded in the cuticular membrane and establishes a primary transpirational barrier. So in addition to sun scald, intracuticular wax can help prevent water loss. So we thought that the wax crystals might scatter the incoming light and thereby reduce the amount of radiation that actually reduces the fruit surface. So to test this idea, we exposed wax, or fruit with high wax and fruit with essentially low wax, the shiny ones here, to high intensity lights. we irradiated the fruit for about an hour and measured fruit temperature sort of indirectly by using a forward-looking infrared camera called a FLIR, or a forward-looking infrared sensor called a FLIR. And you can see on the bottom here that, and then we measured temperature every 10 minutes, the waxy versus the non-waxy. Those images that we took were analyzed, the raw images were analyzed using this program called TheraImage. And we can see here that these darker ones are actually cooler fruit. And you can see just by the naked eye that at 30 minutes and 60 minutes, the high wax ones are cooler than the low wax berries. So that's shown in this graph here. Even after 10 minutes, the low wax fruit are hotter or warming up more quickly than the high wax fruit. And at 60 minutes, that still holds true. So over the entire hour, the high wax fruit maintain a lower temperature. The difference in temperature is only about 2 degrees, which may not sound like much, but even a couple of degrees can make a difference between scald and not scald. So even a couple of degrees difference is important. So in addition to looking at sun scald, we looked at whether or not the intracuticular wax could help prevent desiccation. So to do that, we placed fruit in a heated incubator and measured the percent loss over time. And you can see in this picture at 28 days, the low wax fruit, you could tell are pretty dried out versus the high wax fruit, which are still in pretty good shape. And that is shown in this graph here, where even after 6 days, the low wax fruit start to lose more mass. In other words, they dry out quicker than the high wax fruit. At the end of 28 days, the high wax fruit only lost 9.5% of their mass, while the low wax fruit lost about 20.2% of their mass. So that's a huge difference. So the wax really helps maintain the water in the fruit. We have segregating populations for fruit wax. These were created by Dr. Nikolai Vorsa, the breeder and geneticist at Rutgers. We genotyped those populations using GBS, genotyping by sequencing. QTL analysis showed that we find using QTL analysis, we find a region on chromosome 1 that's associated with the epicuticular wax phenotype. We identified a SNP near the QTL that we were used for markers, for marker development. We also identified a candidate gene. It's fairly far away. It's about a megabase away from the QTL, but it is indeed involved in cuticular wax biosynthesis. It's actually a fatty acid elongase that we're still studying. So we developed what are called CASP markers based on the SNP we identified. CASP stands for competitive allele specific PCR. It's you can look up biosearchtech.com to get a better idea of how CASP works. But basically, there are common reverse primers and allele specific forward primers. And we show here that that those plants that are homozygous for allele 1 tend to have high wax, while those homozygous for allele 2 have very low wax, and of course the heterozygotes are somewhere in between. Our waxes, the waxes, the way we measure those are just by ratings right now, where 0 is very low wax or shiny fruit, and 5 is high wax, these sort of dull, dusty looking fruits. So our marker works pretty well for identifying those that should have high wax. So in conclusion, just for the wax on part, we've selected cranberry fruit with high levels of epicuticular wax that should reduce sun scald as well as the impact of water stress in cranberry. The high and low wax genotypes that were identified were used in crosses to create segregating populations. Those segregating populations were used for QTL analysis based on GBS, genotyping by sequencing, and a SNP-based marker that we developed, a CASP marker near the QTL, is being used currently in our breeding program to select for plants that should have high epicuticular wax cranberries. So now I'm going to switch gears entirely and talk about why we want acid off. The short answer to that question is that we eat too much sugar. If you look at this chart here, published by the USDA Economic Research Service, the recommended maximum amount of sugar that people should be consuming is about 3.9 teaspoons per 1,000 calories. If you look at how much we actually consume, whether you're a child, adult, or regardless of income level, we consume at least twice as much sugar as we should, and significantly more in some cases. So we consume too much sugar. So what does sugar and the acid have to do with in cranberry fruit? Well, the titratable acidity, or TA to Brix ratio, is used to determine the formulation for fruit and juice products. We know that some acid is required for flavor, but the sugar acid ratio is really important in palatability. And the idea is if we have less acid, we need less sugar to get that sugar acid balance that works well. If you look at the amount of acid, or the percent titratable acidity in cranberries, you can see that it's quite high relative to other fruits. Crimson Queen is currently a popular variety, and then these are two different selections that we're working with. As compared to oranges, strawberries, apples, and even blueberries, you can see that those fruit have much lower acids. So cranberry is very high in titratable acidity. So most of the acids in cranberries come from quinic, citric, malic, and benzoic acid. Quinic acid is associated with bitterness. Benzoic acid is associated with a preservative in many foods, and that's something that we're looking at in cranberries as well. But for this talk, I'm going to focus primarily on citric and malic acid. Both of these acids together impart the sour or tart taste that you get when you drink cranberry juice or consume other cranberry products. You can tell that that a lot of sugar is required to balance these acids out, because if you've ever had sugar-free or 100% cranberry juice, you know that stuff is like paint remover. It can be really rough and difficult to consume. By lowering these acids, again, we hope to be able to reduce the amount of sugar needed to make tasty cranberry products. To study the organic or analyze the organic acids in cranberry fruit, fresh fruit are frozen, and then the organic acids are extracted using a basic water extraction procedure. The organic acids that we're looking at right now, malic and citric, as well as benzoic and quinic, are fairly water soluble. They're analyzed by HPLC, and a typical chromatogram is shown at the bottom here: quinic acid, malic acid, citric acid, and benzoic acid. So using those methods, our germplasm collection, or the Rutgers University germplasm collection here at the Marucci Research Center, was screened for genotypes that would be low in organic acids. Nick Vorsa's group identified this one we call NJ91712, which is very low in citric acid. You can see compared to blueberry, it's lower than blueberry, and it's lower than all of these other cranberries that are listed here. Stevens and Crimson Queen are very popular varieties that are grown commercially. So this is extremely low in, relatively speaking, in citric acid. As far as malic acid goes, an accession was identified, NJ9357, that is relatively low in malic acid, and in fact has about half the amount of malic acid as Mullica Queen, which is another popular cranberry variety, and lower than these other cranberry accessions shown in the graph here. So These low acid varieties were crossed. Again, we have segregating populations. The citric acid population we're looking at is CA137. The malic acid populations, we actually have two, CM151 and CM155. These populations were again genotyped using GBS or genotyping by sequencing and analyzed for QTLs or quantitative traits loci associated with acid. We found a QTL for citric acid on chromosome 1 that was very good, and a QTL for malic acid was found on chromosome 4. Again, we analyzed the sequences in and around the QTLs and look for SNPs associated with the trait. I have an example here. We have ones for citric acid, but the example I'm showing here is just for malic acid, where the SNP is CC, TC, or TT, the homozygotes and the heterozygote. We've been able to show that the homozygote TT is very low in malic acid. The homozygote CC is much higher in malic acid. And of course, the heterozygote is somewhere in between. In this case, we used PACE markers or developed PACE markers, which stands for PCR allele competitive extension. It's another type of SNP genotyping assay similar to CASP, where we can distinguish the homozygote CC, the homozygotes TT, and the heterozygotes in the middle, running them on a typical qPCR machine, and where the individual alleles are marked with different dyes. So if you only have dye in one channel, you have homozygotes. If you have dye in both channels, you have the heterozygotes. The PACE assays are actually, I think they're trademarked by a company called 3CR Bio, which is out of the UK. But relatively locally in the United States, IDT, or Integrated DNA Technologies, is sort of the go-between. They will communicate with 3CR Bio and create the primer sequences that you need, and in order to be able to do PACE assays. They work similar to CASP assays, and we've had pretty good luck screening our germplasm using the PACE assays. So the question is, why are markers so important, and why they need to be utilized in our breeding program? And that is because cranberry breeding takes a lot of time, space, and resources. Cranberry, as I mentioned, is a woody perennial. It could take 3 to 5 years from pollination to being able to plant plots out in the field. We collect the seeds. If we do pollinations in the spring, we collect the seeds in the fall. We plant the seeds out the next spring, but it takes a couple of years for those seedlings to grow up and be able to be propagated. Remember, I mentioned that that cultivars are propagated, and of course our breeding plots, when we grow up all the different members of a population, are asexually propagated. So each plot is represents a single genotype. Once they're in the field, they can take another several years to really start producing fruit that can be assayed. So the whole process can take quite a bit of time. This is just a small portion of one of the Rutgers breeding plots. We have something like 10,000 plots in those beds. Each plot is about a 5 by 5 foot square, and you can imagine that growing these up, propagating them, planting them all out, and then maintaining them over several years is very, very time consuming and labor intensive and resource intensive. So by using the markers that I mentioned for both wax, organic acids, and some of the other things we're working on, we can screen seedlings way before they go out to the field. In other words, we could tell if a seedling has the potential, by using our markers, has the potential for being low or high in organic acids, or low or high in wax, low or high in other traits such as fruit rot, and only plant out those that meet our criteria. So it could save a huge amount of space and time. Instead of planting out tens of thousands of seedlings, we can plant out hundreds of seedlings, hopefully, that should exhibit the traits we're interested in. So in conclusion, for the acid off portion of my talk, we've been able to, reducing organic acids in cranberry fruit should allow for reduction in the requirement for added sugar in cranberry products. Low acid cranberry genotypes are identified in our germplasm collection and used in crosses. Those segregating populations were genotyped using GPS, and QTLs identified QTLs for citric and malic acid levels. SNPs within those QTLs were identified and used to design pace markers to be utilized for marker-assisted selection. Now, there are lots of people involved in this project. This is a list of the contributing researchers. Nick Vorso is the breeder and geneticist at Rutgers. He is now very recently retired in December. Jennifer Johnson-Chickalise is our research associate that works very closely with Nick's program. Lindsay Ernwine is my ORISE postdoctoral candidate, and she has recently transitioned to a research associate position with us. Lindsay did most of the waxy work that I presented. Stephanie Fong was a graduate student at Rutgers. She now has her PhD and is working at Driscoll's. She did much of the organic acid work that I talked about. Joe Kawash is our bioinformaticist. It crunches all of our data. Sarah Knowles is a current graduate student with Rutgers, following up on some of the organic acid work. I just listed myself here to show my email address if anyone has any questions for me. And I also have lots of people to thank. So as I mentioned, Lindsay at the time was an ORISE postdoc through the Oak Ridge Institute for Science and Education. Much of the organic acid work was actually started with an SCRI NIFA grant that Nick and I had back in 2017. All the basic HPLC work was done under that grant, but all the marker work was done under our current VacCAP grant, which is listed here. The SCRI NIFA award is 2019. So there are lots of people and lots of grant funding associated with getting these projects done. I wanted to list some of the key references that are associated with the information I presented today. The waxy work was just published a couple weeks ago in BMC Plant Biology. We published our reference genome a couple of years ago in 2022. That was published in PLOS One, and that was used for all of our alignments and QTL analysis and things like that. So the reference genome has been very useful to us. And the organic acid work was published in a couple of different papers that Stephanie wrote back in 2021 and 2020 that were published in Tree Genetics and Genomes. I think I went way too fast, but that's actually the end of my seminar. I'm going to stop sharing. And if we have any questions or comments, I'd be happy to work with you. And I really want to encourage people to email me if you have any questions or comments, things that you want to know or we can talk about. Feel free to contact me, outside the webinar.
Great. Well, thank you so much, Dr. Polashock. That was really interesting. So I invite, you're getting a little.
I know we have lots of time. It was only a half hour.
No, that's all right. So, well, I invite our audience members to place questions in the chat, or you can use the raise hand function. We've got plenty of time for questions. And while we're waiting, Dr. Polashock, I have one. I was really interested in the work that you did on wax. So my question is, are you aware or have you observed any other fruit quality traits that could be impacted for selecting high levels of epicuticular wax?
I didn't mention fruit rot. And fruit rot is a huge problem in New Jersey and Massachusetts, and it's becoming more and more of a problem in other parts of the country. And we think the wax, partly because it imparts that hydrophobic self-cleaning surface, we think high wax can actually reduce fruit rot. We have anecdotal evidence that that's true. We have a selection called Bud's Blues that's very waxy, that is fairly resistant to rot. Unfortunately, the yields of bud blues are not good. They're really small fruit, and it's a low yielding variety. But the wax, it's kind of hard to separate the wax from some of the other things going on. But we think that it does help reduce fruit rot. So there are other useful things that fruit wax, epicuticular wax can do for us.
Great. Well, thank you for elaborating further on that. Looks like we have a question from Rodney Sears. Rodney, feel free to ask your question.
Yeah, thanks. Hi, Jim. Just a question.
Right.
You do the wax assessment just by visual right now. Do you have any ideas, or does anybody else have a quantitative way to measure epicuticular wax?
So I didn't go into the complexities of wax composition, and I am not a biochemist, and it gets way beyond my expertise and understanding. So wax composition as well as wax level can be real important, and that's something that we haven't really looked into yet. But as far as actually measuring wax, we are looking into using our imaging methods such as hyperspectral imaging or to get a more of a quantitative estimate of high versus low wax.
Yeah.
And I didn't present any of that data, but that seems to be working well. So
Okay.
rather than just rate the fruit, we hope to use our hyperspectral imaging system to help get relative quantities for wax. They can be extracted, and people do, but again, it's very complicated, and it's a little bit beyond the expertise of my lab.
Yeah, no, that sounds good. Thanks.
Mm-hmm.
Other questions? We got a couple of positive comments in the chat, but now's a great time for questions if you have any. I think Massimo, looks like you
Yeah,
may.
hey Jim, thank you for the presentation. So on the organic acid, have some crosses started to be made with those mutant, and to understand if it's dominant or recessive trait when you start to make crosses with with material that it's not high, like doesn't have low acid?
Yes. So I didn't present that data. A lot of it is presented in Stephanie's paper. So if you're really interested, I recommend you look up Stephanie's papers. Both the citric and malic acid seem to be Mendelianly inherited. But the very low acid are recessive traits. So they segregated at about 3-to-1. Cranberry, of course, is diploid. They segregate in 3-to-1 with 3 high acid versus 1 low acid for every 4 individuals you look at. So yeah, we have a lot of data on that. I just didn't present it. And of course, there's a lot of interactions, which I didn't talk about either. So if you have really low citric acid, you get some compensation sometimes by malic and other acids. So there are some interactions. Some of our really low malic acid plants are dwarfs. So they produce very tiny leaves and they're tiny plants. But we think that at this point, we think that it's a linked trait. We think we can separate low malic acid from the dwarf phenotype. But we are working on reducing the total acids in general. And one of our concerns going back to fruit rot was if we reduce the acids, we might increase fruit rot. It's unclear how much the organic acids might be associated with fruit rot resistance. But so far, it doesn't seem to be an issue. Our low acid selections don't seem to be any more fruit rot susceptible than our sort of normal or high acids. And I think you called them mutants, and I don't know that I'd necessarily call them mutants. They're just sort of naturally occurring. The ones that we started working with or that Nick identified are naturally occurring variants in the germplasm collection that Nick put together over the years since the 80s when he started here.
Go ahead.
So
Looks like we have
Thank
another
you.
question from Rodney Sears.
Yeah. Oh, yep. I just wanted to sort of follow up on that. So have you guys crossed the low malic and the low citric, trying to get a combination of the two low organic
We
acid?
have, and that's
Yeah.
we have, and that's work that that Sarah Knowles is following up on as part of this grant. She's doing all the HPLC work now, and I think Jennifer is on the line and actually might be. I think she can unmute, and she probably knows more about the current status than I do. So Jen, where are we with the low malic and low citric crosses?
Yeah, we have quite a few populations where we've made crosses between the low malic and low citric, and we're able to screen those in the greenhouse. They produce a few berries, and then we can look at the titratable acidity fairly quickly. And this year we're putting out in the field about 50 very low acid plants into plots so that we can take a look at their performance in the field and have larger samples, and maybe we can start making some juices to get a sense of the palatability of these low acid ones. So we're kind of excited about getting those out to the field. Unfortunately, like Jim said, there is interaction. So when you can make the cross between the low malic and low citric, you don't get quite as low a citric as you would like, because the malic, or I should say, the malic acid increases slightly when you have the low citric. So you're not suppressing the total acid quite as much as we would like to.
Yeah, that's pretty interesting. Actually, maybe as a follow-up, if I can, do we know, and I think probably someone here at Ocean Spray, I'm from Ocean Spray, somebody might know here in the building, but what's the characteristic cranberry flavor? Where does that come from? Is that an organic acid or anything? Do you know?
I believe
Yes.
so. I mean,
Yeah.
Yeah, go ahead, Jen.
Yeah, that's pretty much a combination of the organic acids.
Yeah.
Citric and malic are in higher quantities, so they're influence the flavor more. But,
yeah, I think just from a product development point of view, we'd want to maintain the characteristic cranberry flavor,
right.
and at the same time lower the acid. Yeah.
That's why we're anxious to start making some juices.
Yeah.
Right.
There's
We think it's,
another.
of course, it's all. Go ahead, Jen. I'm sorry.
There's another. Thought I turned my camera on. Yeah.
Yep.
There's another plant that we are finding low total acidity, total titratable acidity, that we're also using in our crosses to try and incorporate kind of the three sources. So, we're trying to utilize all the sources of low acid that we have to further decrease it. And that's why we're anxious to start making some juices so that we can taste the flavor, make sure we're not losing the cranberry flavor.
Okay, thanks.
Right. I think we can sort of maintain that tartness that we all like in cranberry products. But you won't need as much sugar to balance out the tartness, of course, is the goal. Keep the cranberry character and reduce the sugar.
Yep.
So, yep, our product testing will let us know if we're on the right track.
Yeah, good.
We have also looked at sugars to try and increase the amount of sugar in the cranberry, the naturally occurring sugars. There's not quite as much variation there, and so we're not as hopeful on being able to increase that a little bit. The naturally occurring sugars.
Cranberry fruit in general are very low in sugars.
Yeah.
Was something like 3% of the fresh weight.
I think so.
Yeah, there's not a lot of natural sugar in cranberries. If you eat raw cranberries right off the vine or at Thanksgiving time, you can tell there's not a lot of sugar in there.
Oh.
Not very sweet fruits. Looks like they, from a sort of a natural or environmental standpoint, it looks like the fruit dispersal mechanism for cranberries rather than animal consumption is water dispersal because they float. So cranberries seem to have evolved for water dispersal rather than things eating them. And in fact, that as far as I know, people are about the only ones that eat cranberries.
Yeah.
I don't know of any animals that really, we have deer here and all kind of other critters, and I'm not sure if there's too many animals that eat fresh cranberries.
It's called desperation feeding.
Well, what they do feed on is the seeds.
Yep.
When we're out picking
Yep.
in the field, you'll find cranberries that have been cut open and the seeds missing.
Yep.
You'll have little piles of cranberries where the animals have been working on eating out the seeds, the voles
Yep.
and the mice and stuff.
Yeah.
Great. Well,
Jim-
fantastic discussion. Any other questions or comments?
So Jim or Jennifer, any efforts ongoing to look at some candidate genes for those organic acids behind what has been published by in the paper from Sarah, like some RNA-seq analysis between the low and high to start to look at candidate genes?
We have looked at candidate genes, not through RNA-seq at this point, but of course, looking at genes in and around the QTL. And so far, we have not found any characterized genes or genes with putative or known functions in and around the QTLs associated with organic acids. I think there was one acid pump gene that we found that might be a possibility. But that's something that we're still looking into, is the sort of the mechanism behind
Yes.
high and low acid.
Based on what you described, like the fact that there is it's recessive and there is an increase of the other acid, partial increase of the other acid when in the recessive one, it seems that probably it's a loss of a gene. So a gene that was it's synthesizing those specific acid, the function doesn't work anymore, and then if the substrate is the same, basically the substrate is used for the other acids.
Right. Yeah,
but-
certainly there's down regulation of the
Right.
biosynthesis, but of course it's more complicated than that.
Right.
Like I mentioned, there's ion pumps and things like that help control the level of organic acids and things like that in the cells. So it's can go beyond simple biosynthetic pathway. There's regulation involved that's important, and I don't think we understand the whole pathway and the interactions yet.
Thank you.
Hi, Jim. I have a question.
Sure.
I'm wondering if you have any thoughts about the potential for pleiotropy of these locusts with regards to other traits. So you mentioned sun scald or fruit rot as, so I'm wondering if the possibility exists for, say, low acid genotypes to be more susceptible to, say, various fungal pathogens or oomycete pathogens that might affect production.
Right. I think I actually mentioned that. That was one of our concerns, is that the low acid accessions might be more susceptible to fungal pathogens. And so far we have not found that. But it was certainly a concern. We haven't grown them enough to know if, in addition to pathogen susceptibility, if there are any other traits that are affected growing these really low acid fruits. And one of the other things I wanted to look at, and we haven't looked at yet, is other plant parts. So we've of course focused on the fruit because that's ultimately what leads to the products, but we haven't looked at what's going on in the plant yet. We don't know, if those low acid plants have any other, if there are vegetative characteristics associated with the low acid. We don't know that yet. We haven't actually looked yet.
All right. Well,
Did that answer your question, Josh, or did you?
Nope. Yep. You did great. I apologize if I doubled up on your.
Yeah, no problem. No problem.
all right. Well, additional questions for Dr. Polashock?
I believe I'm unmuted here. This is Mike Mainland. Enjoyed your talk, Jim.
And Mike, thank you.
Did you, as you were looking at cultivars and selections of cranberries, did you come up with or see any that were much more susceptible to sun scald than others, independent of wax surfaces? Or
It's something that we planned on doing, but again, I'm going to defer to Jen for comment on that one. Jen, you have to unmute.
Sorry, it's very hard to measure scald. Typically, like when I first started here 20 years ago, we tried to separate, when we were sorting our square foot samples of fruit, separate out scald from rot. And I never felt very confident doing that. I think with scald, you have to catch it right after it happens, because the scald so quickly turns to rot that when you're looking at the berries in late September, October, can't really tell. So perhaps harvesting at an earlier date, right after the scald has happened, you'd be able to get a better sense of that. Usually the scald has a more clearly defined boundary on the berry, whereas with rot, it's not as straight a boundary between the rotten or the sound fruit and the injured skin of the fruit. So it's been a kind of thing that we've struggled with, to somehow quantify scald. Peter Odemans.
Go ahead, Jen.
Peter Odemans is very much interested in scald and the heat issues with fruit. So he's doing some studies with that as well. He has a way to measure the temperature of the berry. Go ahead, Jim.
Yeah, I was just going to mention the color. We know that the color certainly affects scald. Those really dark berries, I guess, absorb more solar radiation and are more susceptible to scald than the lighter berries.
Right.
But aside from color, to get back to your original question, Mike, aside from color and wax, we don't know of any other characteristics right now that affect scald. But it's something that we hope to do. But as Jen mentioned, it's sort of difficult to measure. How do you run an assay for scald? We could certainly do fruit heating assays, but an assay for actual scald is kind of difficult.
The reason I asked the question, released a variety, and this was in the 80s, Cape Fear, and it was very subject to scald. And it typically was berries that were not fully blue yet were most susceptible. And just to kind of verify it, we did some lab oven tests and could induce it with, I don't remember exactly the temperatures we used, something on the order of 115 Fahrenheit, and we could induce it. And it was just considerably more severe on this one cultivar than on anything else that I had seen.
Yeah, if you if that work's been published, Mike, I'd like to look it up and familiarize myself with it.
No, I didn't get it published. We looked at it, and the variety then was pretty much dropped. It looked like it could become a major variety, but it didn't, and it was mainly for that reason. You would have rot then developing fairly soon after you saw the scald, but there's no question it was scald and not a fungus or a pathogen involved.
And we know that cranberry scald is associated with similar internal temperatures that you mentioned for the blueberry. I think about 115 degrees Fahrenheit is when we start to see damage. So I'll talk to Dr. Odemans and we'll see if we could come up with assays to look for accessions that have other that for reasons we might not understand at this point that might affect scald susceptibility.
Did you?
To Jennifer's point with regards to monitoring scald, I'm not sure if folks have thought about this, but you know, the potential of high throughput phenotyping technologies. You know, I don't know what it would cost to build or develop a robot to traverse through the plots, but I'm sure some of those skill sets could be developed by staff or faculty there in Chatsworth to run through or to traverse through those plots and measure that on a relatively regular basis to aid in phenotyping. I don't know how close of an image you'd have to get. It sounds like your plot, you have micro plots, so you'd be taking lots of images, but
yeah. So the short answer is, of course, we are working on that. We know that phenotyping for all of the traits we're interested in is a bottleneck that not only takes a lot of time but a lot of labor. I used to have a picture of all the hand sorting and things like that were necessary for rot to determine rot and things like that. And my lab, as well as the lab of Dr. Jeff Nyhart, he just started with our group. He's USDA research geneticist that just started with us here at the Marucci Center. We are working on in-field high throughput phenotyping methods that include not only RGB but hyperspectral imaging, and we also are getting a robot very soon. So yeah, all those things are in progress. It would be great if we could in-field phenotype for scald as well as a whole bunch of other traits, and that's in progress. We have a small cart that we built, and we're still sort of putting together the parts and the sensors to do what we want to do out in the field.
One thing that's a little bit promising about evaluating scald in the field is it's the berries that are more exposed that are more likely to get it.
Right.
The scald. So that
So
would,
they sit on, yeah, so they're sitting on top of the canopy. But even then, I'm not sure. You know, you mentioned rot versus scald. It'd be difficult for us to tell, I think, using imaging, whether we're looking at scald spots or rot spots. But it's something that we might be able to do. You know, our phenotyping ability is getting better, and our image analysis is getting better. So we might be able to differentiate the two.
I had a question for Dr. Mainland. The blueberry you mentioned was Cape Fear. Is that the variety name?
Yeah.
Yes, it was Cape Fear.
Uh-huh.
And we have material around yet of it, should you want to look at it.
That's
We
a
actually have
Quorum.
Yes, it was no low chill parentage involved in it. It's a true northern high bush.
Yeah, we actually have that here, so we can take a look at it, Jim. It's in our collection.
It sounded familiar to me.
Yep.
So blueberries has a lot of wax on the fruit too. I wonder if that might have been a factor. Delayed
They
wax
certainly
development.
vary. Blueberries certainly vary quite a bit for
Right.
wax on the fruit. There's some varieties that are sort of real shiny, just like cranberries, and some varieties that are very waxy.
Cape Fear was kind of an intermediate. It wasn't one of the real light colored, but had a reasonable amount of bloom on it.
Mm-hmm. It could be the wax composition that's important. I mentioned it's very complicated, and it's something that we haven't looked at, and we'll have to collaborate with a biochemistry lab to look at. But it could be that the wax composition, you know, determines that crystalline structure. And it could be that the crystalline structure between Cape Fear and some of the other blueberries was just different.
Well, we are getting near the end of the hour, so we can take one more question if there is one. If you decide to start to log out now, just want to have a reminder that there will be a survey that is sent out via email. So please take the time to complete that survey. That's really important for our team to get feedback on some of our outreach events. With that, any final question?
Well, let me ask one more yet, if there's time. Have you tried drying any of the low acid cranberries? Do they dry, or would there be more potential for drying versus infusing if you had a low acid cranberry?
I'm not sure I understand the question, Mike. You mean for dried cranberries, like sweetened dried cranberries?
Yes, like blueberries, the higher sugar content, the closer they will dry to the texture of raisins, and lower
Okay.
sugar, they end up being like cinders, very difficult to rehydrate.
I got you. I think the short answer is cranberries are so low in sugar in general that whether they're high acid or low acid, if you dry them out, you just get sort of a crispy little shell. There's just not enough sugar in them to get something like a raisin out of cranberries. So sweetened dried cranberries that you buy in the store, the sweetened part is the important part. They are infused with sugar to make them raisin-like. If you just dry up a cranberry, you just get it's like paper, crispy paper.
Well, thanks. Great program.
Thank you. And thank you all for attending. And I'd like to thank the VacCAP group for the invitation and the collaboration, which has been great.
Well, thank you again, Dr.
Thank
Polashock,
you.
for this
Good, Jim.
wonderful seminar, and we appreciate your time. This will be posted on our
Sound like
website.
Sound like Ibrahim had did somebody have one more quick question? No?
I think it was Massimo talking,
Yeah, it was just
Okay.
me. Thank you.
Okay.
but great job. And thanks again for presenting. It's good to see you, Jim.
Thank you.
Bye-bye.
Take
Have a good
care.
day.
Take care, everybody.